and my presentation is the last presentation, and my presentation is focused on biorefineries. As per over a century, conventional refineries are the workhorses of our economy. They are our economic drivers. Likewise, in the next century, biorefineries are going to play a very key role in economy. They are going to be the economic workhorses. And let us see how nanobiotechnology can be used at the upcoming biorefinery facility for effectively converting biomass resources to biofuels, biochemicals, and biomaterials. As you see here, this is the logo of Savita Medical College where I am working, and the image what you see is of my employer, Shri Dr. N.M. Virayan sir, who has provided me kindly the employment to serve the institute. As you can see here, I will quickly browse through the latest advances in biorefineries. If we search the Web of Science with the keywords, namely biorefineries, integrated biorefineries, biofuels, biorefineries, and biochemicals, these are the number of papers that we get. This shows that the topic of biorefineries is very actively being pursued because it's the economic driver of the upcoming processing facility of biomass. Like a conventional refinery, biorefinery is the place at which biomass is processed and various materials and various products that are useful for sustainable development of human beings can be obtained. Integrated biorefinery. Integrated biorefinery is nothing but the biorefinery will be in combination with renewable energy sources like photovoltaic cells, batteries, wind energy, hydrothermal energy, and other renewable sources of energy, so that the energy needed for the biochemical conversion processes will be substantiated from these natural sources. Integrated biorefinery is the norm of the day, and it is supposed to be economically profitable. America and Brazil and most of the European nations, most of the African nations are well advanced in using first generation feedstock for biofuels production. For example, Brazil uses sugarcane, likewise US uses corn, likewise some African countries use cassava powder and other edible materials for the production of biofuels, especially bioethanol. Our focus and emphasis should be on the use of non-edible sources to avoid food-fuel conflict. Basically, cellulosic material like terrestrial biomass should be used as an ideal feedstock for the production of almost all of the chemicals that are produced at the upcoming biorefinery facility. This is called second generation biofuels. Not only second generation biofuels, we can think of third generation biofuels and fourth generation biofuels also. I told you what is integrated biorefinery, so we'll move forward. And in this slide, I wish to show you the market value of the biotechnological products, and the market value has gone roughly an order of magnitude higher. If you can see here, it was in 1983 5,400, and it is now 11,000. Okay, at least doubled. The biopharmaceuticals and bio-based materials are the norm of the day. So far, we have seen what is a biorefinery, how is it different from the conventional refinery, what are the various countries that are pioneers in the biorefinery technologies. Now we will see what are the various feedstocks that are available at our disposal. I'll try to take as little time as possible because the slides and the content that I need to cover is quite high and large. We can assume, we can comprehend, or we can surmise 4 generations of biomass. The second generation being the trees and the plants that are all around us, basically the cellulosic material, and the third generation is the algal biomass, microalgal biomass or macroalgal biomass, marine microalgae, freshwater microalgae like Chlorella vulgaris, marine macroalgae like Ulva rigida. These algal biomass are rich in either starch or cellulose. They constitute a very potential feedstock for the production of either ethanol or biodiesel. If it were to contain lipids, it can be a very sustainable source for biodiesel. Nowadays, biodiesel is already a marketable product, and bioethanol is already in place of use as a substitute to gasoline and diesel. Yes, what I mean by fourth generation feedstock is we can also think of carbon dioxide being used as C1 feedstock at the upcoming biorefinery facility. Carbon dioxide activation is very difficult because of the high stability, thermodynamic stability of the molecule, and conversion of carbon dioxide that is present in the atmosphere is not so easy, but we can take up the challenge of converting all the greenhouse gases including CO2 and convert them to fuels like methane, synthesis gas, ethylene. Such a strategy will not only reduce the global warming but also provides an alternate pathway for the sustainable production of fuels and chemicals. For example, electrochemical reduction of carbon dioxide using copper nanoparticles as electrocatalyst is one of the potential strategies for the conversion of CO2 selectively either to synthesis gas or to hydrocarbons based on the size of the copper nanoparticles. Not only carbon dioxide, we can think of other feedstock like cyanobacteria, and we can think of plastics, non-biodegradable plastics, even water. These are very sustainable and abundantly available materials all around us, and we have to think how effectively we can convert them to very useful fuels and chemicals using nanobiotechnology. That is my emphasis. The two key words, the two takeaway words from my presentation are biorefineries and nanobiotechnology. What I'm going to give to you is basically there is no substitute to enzymes. Enzymes are wonderful catalysts. They are highly selective. Nitrogenase, for example, nitrogenase can make nitrogen and hydrogen react and produce ammonia at the root nodules, and such phenomena cannot be mimicked at the laboratory. So we have to mimic the natural processes including the activity work of nitrogenase and like that. So if we can think of chemical systems which are performing on a par with chemos enzymes, such systems can make the biorefinery facility highly sustainable, because the problem with enzymes is, the kinetics are often slow. Even though the selectivity is high, the kinetics are often slow, and also regeneration, separation, reusability, cyclability, cost. These are all the difficulties that are associated with enzymes. Everybody know, all biochemists know that these are the conventional problems associated with either the microorganisms or fungus or yeast or cellulase or amylase or catalysis or any other things. So if we can think of developing catalytic systems like, for example, polyoxymetallics, heteropolyacids. These heteropolyacids are exotic solid acid catalysts. They are as strong as 100% sulfuric acid. At the same time, they are very easy and very handy to handle. They are non-corrosive. They will not make the system infrastructure corrode. They perform as selectively as that of enzymes. These are only some examples. Polyoxymetallics or heteropolyacids are such exotic metal oxygen clusters developed by J.B. Moffat and many other Japanese scientists, including the Russian scientist Kozhavnikov, and many other scientists have done pioneering work on polyoxymetallics. So what we have to think, what should be our approach for the sustainability of biorefineries is that we should develop chemical catalysts that perform on a par with enzymes. At the same time, the classical problems associated with these enzymes, fungus, bacteria, microorganisms should be overcome. For example, polyoxymetallics and heteropolyacids. Also, very recently, Professor Arun Gedankancer's group from Israel has developed a magic material called zinc doped copper oxide. Copper oxide we know very well. If copper 2 plus ions are selectively replaced by zinc 2 plus up to 12.8%, then what we get is we get beautiful magic material called zinc doped copper oxide, and this zinc doped copper oxide has been tested for various applications, and not only Professor Gedenkenser's lab, all over the world many research groups have tested the unique features of zinc doped copper oxides, and they found that these simple metal oxide nanoparticles, these are nanoparticles, of course these are not bulk, they are highly potential antibacterial materials. They are very highly potential antifungal materials, means they kill microorganisms that can cause harm to human health. So how this happens, I come back to that. Okay, now we are at the various generations of feedstocks, and you can see here the image. What you see is the typical representation of lignocellulosic materials. The green wire like things what we saw are hemicellulose, and the brown thread like things what you see is lignin, and the cylinder, green color cylinders inside is cellulose. Cellulose is highly crystalline, and it is a non-aromatic polymer of glucose, and the hydrolysis of cellulose under microwave irradiation in the presence of acid catalyst like heteropolyacid gives selectively glucose, and glucose is very vital feedstock, and in fact glucose can be converted to various other chemicals and feedstocks at the biorefinery facility, because the product diversification is very much possible with glucose. Glucose can be isomerized to fructose. Fructose can be dehydrated to HMF, hydroxymethyl furfural. Hydroxymethyl furfural can be again rehydrated and then dehydrated in the next step to get levulinic acid. Levulinic acid is a wonderful bioplatform chemical, because it has two functional groups. One is carboxylic group and carbonyl group. The moment we have many functional groups, functionality is like hands. I do have two hands, okay, so I can perform two functions at a time. I can take my water bottle, I can control my mouse. Likewise, if I have 10 hands, the functionality is going to tremendously increase. When the functional groups in the molecule are increased, its reactivity increases, its diversification increases. Levulinic acid is one such molecule. Glycerol is one such molecule. Glycerol has 3 hydroxyl groups. Likewise, furfural has 2 functional groups, hydroxyl group and the aldehyde group. So when we have such biomolecules obtained from biomass, what will happen is they can be diversified. A variety of chemicals that can be produced from conventional refinery can also be produced at the biorefinery facility just by changing the catalyst, just by changing the environment, whether it is acidic, whether it is basic, whether it is neutral, whether we are using metal oxide, whether we are using acid catalyst, basic catalyst. There are many ways in which chemists can control the processes, the selectivity, the yield, all these things at the molecular level. Catalysis science has developed so much, and the knowledge of catalysis that has been extended so far to conventional biorefinery should be extrapolated to biorefinery facility, for the biorefinery facility to be performing on a par economically with the conventional biorefinery. The pie diagram, what you see is the relative amounts of cellulose, hemicellulose, lignin, and others. And cellulose is the major component of lignoterrestrial materials, lignocellulosic terrestrial materials. After cellulose, the other major components include hemicellulose and lignin. They are 30% each. However, cellulose is can be around 40% or so. What I mean to say is cellulose is the single largest carbon non-aromatic material present on the surface of the earth that can be a feedstock for glucose followed by bioethanol. Okay, if we want to go for biodiesel, we should go for other feedstock. We should not go for carbohydrate. Carbohydrate cannot be used for transesterification reaction, for reacting with the methanol and yielding fatty acid, amyl ester, fatty acid, alkyl ester, or glycerol. We have to go for feedstocks like oils, lipids, vegetable oil, for example. Every day when we cook at our home, we generate enormous amount of waste cooking oil. The waste cooking oil can be reacted with methanol under non-conventional heating sources like microwave irradiation, and the transesterification process can be accelerated several orders of magnitude. Microwave irradiation is such a unconventional heating technique wherein the heat spots are generated. The heating method is very different compared to the convective oven. So these are all the possible ways that are available at the dispensation of the chemical engineers and chemists to have the demand-based supply of either ethanol, biodiesel, or gamma-valorolactone or methyl tetrahydrofuran. Not only this, the current interest or the current focus of the biorefinery is green hydrogen. How to produce green hydrogen? There is no substitute to water electrolysis. Once we have the electrolysis of water, we have hydrogen ready. But how to make the hydrogen production greener means the electricity that is used in the electrolyzer should come from renewable energy sources like photovoltaic cells and biomass and other renewable sources, and then the hydrogen is called green hydrogen. The time is very limited and I'm running short of time, so I'll quickly show you. These are various platforms available at the biorefinery facility, the conventional sugar-based platform and the syngas platform. Apart from syngas platform and the uh, chemical platform, we can think of two other platforms like electrolysis of hydrogen, electrochemistry, okay, electrocatalytic reduction of carbon dioxide. These can be thought of different platforms, because here we think of sugars, we think of carbohydrates, but we do not consider water, we do not consider plastics, we do not consider CO2. Such chemicals also should be considered, and other platforms are to be developed at the biorefinery facility. The extreme right, what you see is various bio feedstocks for the production of long chain alkylamine, that is biodiesel, and then phenol. Phenol is from aromatic polymer, that is lignin. This is called hydroxymethyl selective production of hydroxymethyl, which is called the sleeping giant. It has many applications in pharmaceutical industry and food industry. This is glucose, and this is glycerol, and this is ethanol. These are all the various molecules that can be obtained from biomass conversion. The energy density of ethanol is 10 units less than gasoline, and it's greener fuel. Sustainability is one important aspect that we can attain from the biomass, and Professor Nasib Qureshi has defined the sustainability as like this. Sustainability is a system of production processes for which the life cycle assessment suggests preservation of biomass production capabilities like the land fertility, water resource, CO2 sink capabilities, and biodiversity, and the minimization of the use of fossil-based fuels. This is the famous Professor Qureshi from National Agricultural Research Center, US Department of Agriculture. And then I'll move quickly to a process called fermentation. You all are very familiar with what fermentation is. The problem with fermentation is conventionally fermentation is a very, very slow process. What we have developed is we have developed a solar-based reactor to do the fermentation process at a very high rate. Not only that, the problem with fermentation process is there will be substrate poisoning, there will be a product poisoning. You know, when ethanol concentration in the broth is more than 4%, there will be product poisoning. Whereas in the system that we have developed, the beauty is the moment the ethanol that is formed is removed from the system. So always the ethanol, the yeast that is present in the fermenter feels the presence of sucrose or glucose, so there will not be any product poisoning, and the formed ethanol will be evaporated and condensed on the top walls of the solar reactor that you are seeing here, and then collected on at the another collection chamber, and from the second chamber wherein the ethanol is collected, the ethanol will be collected, and then the ethanol can be directly used as a fuel for direct ethanol fuel cells. You will be surprised the concentration of ethanol that we can get in this solar assisted reactor is nearly 19.2 weight%. Means if I feed 28% glucose, what I can, if I feed 48% can get nearly half the concentration of glucose will be the concentration of ethanol, because we all know that fermentation of sugars is 50% efficient at bioethanol producing plants are a rich source of uh, carbon dioxide as well, and that carbon dioxide need to be converted by non-catalytic and electrocatalytic, and catalytic means to syngas and fuels like methane and uh, ethylene. Okay, moving forward, this is the schmarine, that is yeast. Glucose is metabolized by schmarine to give 2 moles of ethanol and 2 moles of CO2. It's not in principle that simple. There will be many secondary metabolites, especially as with the transesterification of fatty acids and lipids. Glycerol formation is inevitable in fermentation process. Also, glycerol is the secondary metabolite. We have found that the yeast will also metabolize the glycerol and convert it into 1,3-propanediol. 1,3-propanediol is a precursor to polymers. It's a very beautiful starting material for a very many polymers. Okay, so the glycerol that is considered as a waste stream in the biorefineries, for example, in the bioethanol production plant or biodiesel production plant, can be successfully converted to 1,3-propanediol, which will be a promising feedstock for the production of polymers. For example, platinum-based catalysts are used for the
Sorry for
conversion of
interrupting you,
yes.
Finish.
Here is a nano yeast being generated and supported on silica. Such a nano yeast particle, if they were to be coated on activated carbon, functionalized with carbon nanodots, they will have a high surface area and many active sites wherein the small nanoparticles of yeast could be absorbed. And in sun, if we do the fermentation reaction, the rates will be higher. Okay, I'll move forward. This is the proton NMR showing the presence of ethanol. Product characterization is very important because conventionally different methods are used for the characterization of fermentation product. With the advancement in analytical techniques, you know what were not known or characterized previously, what the missing links can be connected, and we can find products like 1,3-propanediol. Based on the HPLC and NMR analysis, we have shown that fermentation broth also contain 1,3-propanediol, and it needs time and effort. We allowed the fermentation process for 3 months in the solar reactor, and at the third month, we have collected the residual uh, material in the fermentation broth and analyzed and found that it has 1,3-propanediol. Okay, so the knowledge that was not known few decades ago can be known with the advancement in the technologies, analytical techniques that we have. So all is not very known and very clear to us. Fermentation, though it is a century-old chemistry, much needs to be explored. Okay, moving forward, nano yeast for bioethanol production. You can see here the small egg-like features, 40 nanometer nano bud. Yeasts, we know very well, it's the smallest size we can think of is 4 to 5 micrometers, but I'm talking about nano species of yeast. It's possible. We used a special sonication technique to develop a nano yeast, and you see this is the bigger mother yeast, and mother yeast has buds, and these buds can be removed by sonication process, and they can be adsorbed on support, and such orphaned yeast nanoparticles are supposed to perform exceptionally well for fermentation reaction. This is again 1H-NMR. The Department of Energy, US Department of Energy has identified 12 chemicals to be the starting materials and building blocks for a variety of chemicals that can be generated at the biorefinery facilities. And all these 12 chemicals has one characteristic thing. They have oxen containing functional groups, nitrogen containing functional groups, either oxen or nitrogen or both. So that's what I said. The greater the functionality, greater is the chance to get converted to various products that are useful for various industries. Moving forward, green synthesis of furfural from xylose. The beauty of this particular work is that here we have used zinc-doped copper oxide, such a very simple nanomaterial, for the selective dehydration of xylose to furfural. The same thing can be used for the selective dehydration of glucose to HMF as well, and such work is in progress. And remember, zinc doped copper oxide is such an exceptional nanomaterial. It can have diverse applications. It can have many applications, including antibacterial activity, antimicrobial activity, antiviral activity, antifungal activity, and catalytic activity as well. Okay, moving forward, yeah, this is the simple reaction. Xylose gives 1 mole of furfural and 3 moles of water molecules, and our zinc doped copper oxide is doing exceptionally well with 86% yield, and this is the mechanism. The number of active sites are increased when zinc is present along with Zn Cu2 plus, and these are the amounts of the desired product that are formed with the zinc doped copper oxide. The black color bar is the furfural, and these are the 13C NMR and proton NMR proving the presence of furfural. And the 4 characteristic signals in proton NMR in the range of 6 to 10 are symbolic of furfural, and analysis of product is very important. So I'm just trying to drive home the point that NMR is one elegant technique where any kind of sample can be put it in the machine and we can get new information. Very simple and handy, unlike HPLC and GCMS and MALDI and many other things. Okay, then I want to introduce to some of the audience who are interested solid acid catalysts. These are Kegin type polyoxymetalates. You see the Kegin structure. This is very characteristic 12 MO6 octahedra forms this Kegin type structure, and this is very famous material in the chemistry of hydropolyacids. And Kegin did X-ray crystallographic studies and revealed the structure of the material and the position of the protons and all those things. H3PW12O40, H4SiW12O40, cesium 2.5, H.5 PW12O40. These are various kinds of heteropolyacids with a graded acidity, and then they can be used for the selective hydrolysis of cellulose to glucose, which is the key economic driver for the upcoming biorefinery facility. If the silicotungstic acid is supported on activated carbon, you can see we can get only exclusively glucose. You see the 8 peak patterns that you see here are the glucose, and with increase in temperature, we can see that the starch that is taken as a substrate is completely converted to glucose. And the heteropolyacid supported activated carbon has been used for the starch hydrolysis reaction for at least 5 cycles, and more than 90% yield is obtained, and this is a selective catalyst for the conversion of either cellulose or starch to glucose. That's what I said. We are trying to find enzymatic mimics. We are trying to find a substitute to cellulase and amylase. You see, amylase is used for the conversion of starch to glucose. Cellulase is used for the conversion of cellulose to glucose. The same thing we can do with heteropolyacids when it is supported on activated carbon and graphene. In fact, we have shown that the bare heteropolyacid is 8 times lower in activity when it is the same material when it is supported on graphene and used for the hydrolysis of glycogen. Its activity is tremendously enhanced because of the increase in the number of active sites. Moving forward, this is the glycogen, and glycogen converted to glucose using a microwave and sonication and hydrothermal process. Initially, HCl is used. We found that HPA is promising a substitute to hydrochloric acid, and phosphorus 31 NM, 13C NMR is shown here. Phosphorus 31 NMR is to show the stability of the Kegin type polyanion even after the reaction. The honeycomb lattice, what you see is graphene, and the blue color dot-like materials, what you saw are polyoxymetalates, that is Kegin type polyanion. When such Kegin type polyanion is supported on graphene, you see the activity is enhanced by 8 times. And we can think of simple materials like pine cones and convert them to glucose, and even the xylose component can be metabolized by yeast, and we can get ethanol. And we can think of marine macroalgae like Ulvarigida and convert them to as high as 16% ethanol under sonication conditions, and carbohydrate rich algae can also be produced. And this is the typical solar reactor that I was talking of that has resulted in as high as 19.28% ethanol from 48% glucose, and these are the 13C and proton NMR spectra, and the product ethanol was used in fuel cells, and the fuel cell performed exceptionally well. And this is the continuous flow process for the production of high ethanol concentrations. And moving forward, 1,3-propanediol can be produced by the metabolism of glycerol by yeast, and Okay, these are the various diagrams showing how we can use various lignocellulosic agricultural biowaste for the production of levulinic acid. These are different lignocellulosic materials we have used for the production of levulinic acid. Why levulinic acid? Levulinic acid can be a feedstock for starting material, what to say, platform chemical for fuel grade chemicals like gamma valeriolactone and MTHF, methyl tetrahydrofuran. These are called aviation fuels. Those are called aviation fuels. And moving forward, this is the work I did in Korea in the laboratory of Professor Taehun Kim sir after winning the Advanced Scientific Brain Fellowship in South Korea. And this is the microwave irradiation of glycerol, wherein fast production of glycerol levulinic acid can be obtained, and it is because of the synergistic effect of zinc bromide and HCl, wherein hydrogen bromide is formed. And these are modified heteropolyacids, gallium modified heteropolyacids, where there is an optimum Lewis and Bronsted acidities, resulting in the selective production of levulinic acid from glucose. And these are elemental maps. These are scanning electron microscopy with elemental mapping. This is the trend in which biodiesel is investigated. Biodiesel is produced from cooking oil, and strontium oxide is used as a solid base catalyst. And quickly moving forward, carbon dioxide can be converted to fuels like hydrogen formic acid, and then synthesis gas and others. And these days, active research is on how to produce green hydrogen using nickel-based systems. Okay, nickel oxide with in combination with graphitic carbon nitride and many other systems, nickel oxide, nickel oxide platinum, and all these electrocatalysts were generated for the selective production of hydrogen from photochemical or photoelectrochemical or electrochemical means. And these are all the pioneering works that have been produced during the last 5 years. And majority of the work deals with non-noble metal catalysts like yeah, here this is exceptional. You see John Teller distortion because of the presence of nickel 3 plus. Such defects are very crucial for the enhanced activity for the production of yeah, you see, doped systems, the energy is minimized, the free energy is minimized, doped nickel oxide, and such doped systems are performing much better compared to yeah, on a par with noble metal catalysts for the photoelectrochemical and electrochemical splitting of water to hydrogen. And yes, let me complete. Okay, these are the plastic ways how plastic can be converted to fuels and chemicals. This is the market size of biofuels. These are the pioneers in the industry. These are the various players in the industry. Recent literature on the biorefineries, what is the market size, and what are the latest developments. And I wish to conclude by giving thanks to my supervisors. A concluding remark: there is plenty of room at the upcoming biorefinery facility and biochemical industries. Solid acids and solid waste catalysts opened a new avenue for as a substitute to biocatalysts like amylases, catalases, and cellulases. And I wish to conclude my presentation by acknowledging my teachers, my PhD supervisor, Professor Tirukalan Kantad Varadarajan sir, and my PhD co-supervisor, Professor B. Vishnathan sir, for giving me the fundamental knowledge on performing scientific research. I am indebted to my postdoctoral supervisor, Professor Aharon Gedenkan sir, for giving me a blessed opportunity to be in Israel for 6 long years. And most of the work that I have presented here is the work that is conducted in the laboratory of Professor Aharon Gedenkan. That is why he has been a co-author in the presentation of this lecture. And I'm grateful to Professor Tehun Kim sir, who has been instrumental in securing me the award for Advanced Brain Pool Fellowship. I'm thankful to Professor Xinling Wang sir from Shanghai Jiutang University, who has given me 2 years of fellowship in China, who has enriched me with knowledge in carbon fiber reinforced composite. Chongchong Yang, Dandan Ju, Zhenjiang, Professor Thomas sir for giving me the professor visiting professor fellowship in Mahatma Gandhi University, MSK, MS Shrikala madam, and then Uday Bhaskar Tumu for being a good collaborator. I am grateful to the former librarian Jadhav sir for providing me very calm environment, and the current in charge of the Library, IIT Madras, Muruganandam sir, for providing me all the needed resources and facilities for conducting scholarly works, for writing books, for delivering lectures like this. I'm thankful to Robinson sir and Karuna Karan sir and Professor G. Shekhar sir, Damini, for giving me many opportunities to share my knowledge with researchers in the field through NSTC. I'm thankful to Karshit Bhaskar. I'm thankful to teacher Ankita Srivastava, who had opened before me the door to present my lectures in NSTC. I'm thankful to my friend and colleague, Professor Trupti Gajaria Madam at GSFC University, for being highly inspirational and motivating, and for being a wonderful friend and guide. I'm thankful to Professor V. Kamakuti sir, the director of IIT Madras, for opening the door of IIT Madras and offering the facilities for even to the least of the researchers to access the facilities at IIT Madras. I'm indebted to Professor Dr. N.M. Veerayan sir, the founder and chancellor of the Savita Deemed University, and I'm greatly indebted to Jennifer Landers for giving me this wonderful opportunity to present my work before you. And she has been an exceptional friend to me, a wonderful friend to me. Thank you, Jenny. And last but not the least, I'm thankful to my Lord and Savior Jesus Christ for empowering me with knowledge and wisdom and power to deliver my best. I am associated to all these blessed universities. May God bless all these universities. And thankful and thanks are due to all the audience for their august presence. Thank you.